It is my pleasure to introduce Nicola Jones to you. She'll be speaking to us today, Pediatric Grand Rounds. Nicola Jones comes from the Hospital for Sick Children in Toronto, where she's a professor of pediatrics and physiology and a senior scientist at the Research Institute in Cell Biology. Her career started much more in the medical field, but as I'll explain, she really evolved to be a key figure in the research and academic world. So she started her undergrad at Queen's and then was at the University of Toronto for medical school, pediatric residency at Sick Kids, and GI fellowship at Sick Kids. And then, similar to the path that I then followed, she discovered the lab during her fellowship and ended up doing a PhD with Philip Sherman in cellular microbiology, focusing on Helicobacter pylori, which she still has a very keen interest in. And at that time started to build her not only personal interest, but her reputation, which has now really gained national and international recognition in understanding Helicobacter pylori initially, but other biological processes, including autophagy, which you'll hear about today. During the time that she's been at Sick Kids, she was program director when I was there. So it really is great to have her hosted here by the division. But she has also carried many key roles in national and international bodies related to research, education, and academic development, including being the head of the research committee for the Crohn's Colitis Foundation of Canada. She's had key roles in the Canadian Association of Gastroenterology and the North American Society of Pediatric Gastroenterology, Hepatology and Nutrition, where she sits on the executive. And probably one of the most important appointments that she's had recently, which is relevant to many people here, is she's the director, principal investigator of the CCH-CSP, the Canadian Child Health Clinician Scientist Program, which does identify individuals at different levels of training who are interested in either becoming or enhancing their clinician scientists training and becoming clinician scientists. I was involved in that program when I was over at Sick Kids, and it really is a great opportunity, so I encourage anyone who has interest in that area to speak to Nicola or our leaders over here. And she, of course, throughout this time has published in key journals and really has given talks all over the world. So really it's a pleasure to have you here, Nicola, today to speak to us. So please welcome Nicola Jones.
Well, thank you very much, A10. That was a really kind introduction, and it's been a real pleasure for me to be here. I've been here for the past few days, visiting with lots of people and giving some talks, and I've been very impressed by the environment here. It's a great place, so it's nice for me to see A10 doing well in such a great place. So I'm a little concerned that the presentation next week is the resident skit, so I'll try to be as entertaining, but I'm not sure I can guarantee. So the title of my talk is Healthy Eating and Gastrointestinal Disease. And so with the title, and I saw the advertisement, I think there was carrots on the image. So some of you may be thinking, oh, you know, she's coming to talk to us. The holiday season is approaching, departmental holiday party coming up. Maybe Sue wants you to, you know, practice a little discretion. You know, let's not go overboard. I'm going to give you tips for that. Or, you know, could it be, please tell me it's not so, but are we already talking about our New Year's resolutions and how we're going to eat healthy moving forward? So in fact, none of that is true. I am not going to talk about that kind of eating. I'm going to talk about a different kind of eating, which is eating within a cell. And this is a process called autophagy. So just show of hands, how many people have heard of autophagy? Okay, excellent. So quite a few of you. So that's great. So if you haven't, don't worry. I hopefully will give you an understanding of what autophagy is. Either somebody's got really high heels and they're coming down the stairs, or there's construction going on. I'm not sure. So I'm going to talk to you about some work that we've done looking at the role of autophagy in gastrointestinal diseases. And I'm going to focus on two diseases, Crohn's disease and infection with Helicobacter pylori, which is my favorite organism. And then in the end, we're just going to talk about some of the potential therapeutic implications. So I thought that I would start off with a case to really put this in focus and why we're interested in this. So this is a case that we would see in our gastroenterology clinics, or you might even see this in your general pediatric clinics. So it's a 9-year-old boy who presents with a history of poor growth, weight loss, abdominal pain, and loose stools. And importantly, his family history is positive. His mom has inflammatory bowel disease. So with a presentation like this, what we would do in the GI clinic is we do some labs, obviously physical examination, and we do an endoscopy. And what you can see on endoscopy here is a very inflamed bowel, and we can also see some areas that are normal. And when we take a biopsy, so this is a biopsy of the intestinal mucosa, we're looking at this under the microscope, and we see this characteristic granuloma. So for us, this is diagnostic of a child that's got Crohn's disease. So why does this child have this disease? So what's our current understanding of how inflammatory bowel disease is caused? Well, really, this is a schematic representation of years of research and lots of genetic studies that have really highlighted what are the important factors that result in predisposition to inflammatory bowel disease. So we know that there are host genetic factors that are involved, and we think that these host genetic factors impact on the mucosal barrier so that you can have defects in mucosal barrier. They also alter the host immune response, and it's clear that there's a major role for microbes. And so what we think is that the host responds inappropriately to gastrointestinal microbes. There's also a role for environmental factors, and all of this multifactorial features result in the development of inflammatory bowel disease. So that's what we know at present. So I'm going to tell you about our work that's focused on looking at the role of bacteria and this autophagy pathway in disease. So what's the role that bacteria or microbes are involved in inflammatory bowel disease? Well, there's actually quite a lot of evidence. So in animal models of inflammatory bowel disease, if animals are raised in a germ-free environment so that they don't have any colonization with microbes, they don't develop inflammation. We know in patients, so we sometimes treat patients with antibiotics and it can reduce symptoms. If we look at the tissue from patients with inflammatory bowel disease, we can see that bacteria more commonly penetrate the mucus layer. And then finally, if we look at animal models of inflammatory bowel disease, we can actually transfer inflammatory bowel disease from one animal to another by transferring their microbiome. And then, importantly, the genetic studies looking at what are susceptibility genes in inflammatory bowel disease have really highlighted the role of host microbial interactions. So just published very recently was a meta-GWAS, where basically they took all the genome-wide association studies, put them together like a meta-analysis, and identified that the significant pathways involved in inflammatory bowel disease were implicated in host microbial interactions. So really lots of evidence to suggest that the microbes are involved. And so many of you will know there's a huge interest in studying the microbiome. And so you can see up on the left the microbes lining the mucosa of the gastrointestinal tract. And in fact, many of you will have heard this many times now, we are more microbes than we are human cells. And so I like to think of us really as condominiums for our microorganisms. So with respect to inflammatory bowel disease, people have been looking for the smoking gun. So is there a specific pathogen that causes inflammatory bowel disease? So is there a bad bug, or is it really that our good bugs have actually gone bad? And I can tell you with the discovery of H. pylori and its role in peptic ulcer disease, that really spurred on people's interest in could we find a pathogenic organism that causes inflammatory bowel disease. And so far we haven't found that. So instead what we think is that it is really an alteration in our good bugs. And so this is a slide which is really a summary of some work by Dan Frank's group that's looked at the difference in the microbiome in the intestine in normal individuals and those with inflammatory bowel disease. And it's summarized in this figure. And so you don't need to worry about specifically what are the bacteria groups at the bottom, but each color represents a different phyla. And so if we look at a normal individual, this is from tissue going from the ileum all the way down to the rectum and their feces, and looking at the proportion of these specific families of bacteria. Now, when we contrast that to individuals with inflammatory bowel disease, you can clearly see the pattern is different. So there's a change in their normal flora, and we call this dysbiosis. So it's clear there's dysbiosis in patients with inflammatory bowel disease, and this has been shown repeatedly now. So the question is, why is there this dysbiosis? So if we look at the intestine here, so there's the epithelial cells, here's our lumen with our commensal organisms, maybe some pathogens. So why is it that we would have this dysbiosis? Is there some way that we're not regulating properly our commensal microflora? And so I'm going to tell you about some of the work that we've done to show that, in fact, this may be the case. So these were some of the very first susceptibility genes identified associated with Crohn's disease, and actually a multigenic disorder. So these were really seminal discoveries. And they came from GWAS, but also from candidate gene studies, and identified this gene called NOD2, which is associated with Crohn's disease. So just another show of hands, who knows about NOD-like receptors? Okay, so less know about NOD-like receptors, more know about autophagy. Okay, so how about toll-like receptors? Who knows about toll-like receptors? Okay, more. All right, so this is my schematic representation of a toll-like receptor. So here is your cell, and on the surface of your cell are all these receptors which sense microbes and microbial components. Those are the toll-like receptors. So if we go within the cell, then these are the NOD-like receptors. So these are receptors within the cell that sense bacterial components. So this discovery that alterations in NOD2 were associated with inflammatory bowel disease, with Crohn's disease, suggested that maybe somehow there's a change in how we sense the bacteria. So this is just a diagram showing you what NOD2 does. So here we have a cell, here's a bacteria, and here's our NOD2 receptor in the cell. So when a bacteria infects a cell, it releases these components called peptidoglycan, and these are parts of the cell wall of the bacteria. So when a bacteria infects a cell, NOD2 recognizes this peptidoglycan, and it sets off a cascade of signaling that tells the cell, oh, there's infection here, I've got to respond to this. So that's the job of NOD2. So this is NOD2. So it has several different regions to the protein. And the most common mutation associated with Crohn's disease results in the loss of this end bit of NOD2, and it loses its ability to sense that component of the bacterial cell wall. So this is just some results from one of our collaborators, Stéphane Girardin, showing this effect in patients' samples, so blood cells from patients that are either wild type for NOD2 or they have the Crohn's disease variant, and you can see that in the hashed line. So here's untreated cells. When we add that component of the bacterial cell wall to normal So individuals that have normal NOD2, the cell says, whoa, infection, let's do something, and it responds by activating an inflammation cascade. So interestingly, in patients that have this NOD2 frame shift, where they don't recognize that peptidoglycan, they don't have this inflammatory response. So if you think about it, this is kind of strange, right? So why would decreased bacterial sensing increase an inflammatory response in Crohn's disease? And so that was really a question that people couldn't understand, and we were trying to figure out how could NOD2 be involved in this inflammation response. And then these papers came out. So further genome-wide association studies, and they identified this gene, ATG16L1, a variant in that gene to be associated with inflammatory bowel disease. So this actually took everyone by surprise. So nobody was thinking about autophagy when they were thinking about inflammatory bowel disease. So it shows you the power of genome-wide association studies, because really you have no hypothesis going in. You study the genes of a whole bunch of individuals that either do or don't have the disease, and identified that this pathway was associated with having inflammatory bowel disease. So we were really excited about this. And so let me just tell you a little bit about ATG16 and autophagy pathway, and then our work that's been looking at this. So this is ATG16, and here's where the single nucleotide polymorphism is in ATG16. And it results in an amino acid change, and exactly how that affects the function of the protein isn't really clear, but I'll show you some results that we have to suggest how it might impact. So it's actually quite a common polymorphism. So half of the Caucasian population carry a copy of this polymorphism. So it's very common. And individuals with Crohn's disease, there's a slight increase in the proportion that carry this allele. So if we look at the GWAS studies or the meta-analysis, having this variant increases your odds ratio for developing Crohn's disease by about 1.5-fold. So a modest increased risk for inducing inflammatory bowel disease. So what is autophagy? So very literally, autophagy means eating oneself. And I always love showing this slide in a pediatric audience because we can all completely relate to it. And the other important point I just want to make is this is not my son. So what is this pathway? So initially it was identified in cells as a cellular recycling pathway. So when a cell was under a condition of starvation or nutrient stress, it would start to self-cannibalize itself, and it could then use those nutrients, recycle those nutrients to allow the cell to have energy and survive a stress like that. So this is basically what happens during the autophagy pathway. So this is within a cell. Here are some organelles in the cell. And if there's a trigger to undergo autophagy, then this isolation membrane forms. It starts to engulf the material in the cytoplasm and eventually forms this double membrane vacuole. It then fuses with the lysosome, and the lysosome's got degradative enzymes, and so that results in the degradation of the material within the autophagosome, which can then be recycled and used in the cell. So there are two pathways that are important to know about in autophagy. So one is the ATG5, ATG12, ATG16 complex, which is shown here, these little red dots. So when there's a trigger to undergo autophagy, these proteins associate with each other, and they're important for where this isolation membrane is going to form and for completion of autophagy. So it's absolutely necessary. If you don't have these, you don't have autophagy. The second pathway involves this protein called LC3. So normally, you can see it in yellow here, LC3 is found in the cytoplasm, and when there's a trigger to undergo autophagy, it gets modified and lipidated so that it will then go to the isolation membrane, and it stays with the autophagosome until the contents are degraded. So importantly, we can identify autophagy in cells by tagging this LC3 with a fluorescent probe that then we can follow when we look at cells under the microscope. Also, importantly, when we take cells and we lyse them and we run them on a gel, we can identify autophagy because that change in LC3 makes it migrate at a different rate, so we can actually see it on a Western blot as well. So different ways that we can identify autophagy in cells. So originally it was thought this was the importance of autophagy, but it's now clear that it has much more important roles. It's very important in immunity. So this is a schematic representation of some of the roles that we've identified with the autophagy pathway. So again, here's a cell. Autophagy is important for getting rid of intracellular pathogens. So here's a pathogen coming into the cell, and it may be within the cell in a vacuole or free in the cytosol. And autophagy will recognize that bacteria, ingest it, and help to degrade it and get rid of it. So autophagy helps control infection. Autophagy is also important for activating the innate immune response. So I told you about toll-like receptors. Remember the bus with the people on the top? So somehow, and we don't know exactly how, autophagy is also important for activation of some of these toll-like receptors. And then finally, autophagy is also important for antigen presentation. So peptides are present within the cell. They're ingested by an autophagosome and loaded onto the MHC class molecules, which then present the antigen, which allows adaptive immune response as well. So you can see it's much more than just a cell recycling process. It has a lot of additional roles. So what could be the potential role of this protein in inflammatory bowel disease? Well, when we started our work, there were 3 groups that had done some studies to look at what could be the potential role. So this is a normal epithelial cell lining. And here at the bottom we have something called Panus cells. These cells secrete molecules which help to control infection. We have immune cells, and here's a bacteria that's come into the cell and autophagy's ingesting it. So the first group tried to develop an autophagy deficient mouse, but this mouse doesn't survive. And so what they did was they took immune cells from the fetal liver, and they were able to culture these cells. When they added a bacterial component to those cells, the cells that didn't have proper autophagy, there was an increase in an inflammatory response. So suggesting that maybe autophagy helps to control the inflammatory response. But remember, these are completely autophagy deficient cells, so not exactly the same thing as the Crohn's disease variant. Another group developed an animal model as well. So they developed a mouse, and this mouse had less ATG16 expression, so 30% of what the wild-type mice would have. So again, it's not exactly the same as the Crohn's disease variant where there's an amino acid change, but it may be similar. And interestingly, what they found was that these Paneth cells, which secrete molecules to help regulate infection in organisms, they weren't secreted properly. So instead of these nice little secreted vesicles, they were all abnormal. And interestingly, when they looked at tissue from patients with inflammatory bowel disease that had the Crohn's disease variant, they also had this change in the Paneth cells. Now, interestingly, in the mouse, this hypomorph mouse, it only had these Paneth cell abnormalities if it also had a viral infection. So suggesting that maybe there's an interplay between infection and the ATG16 and its effects. And then finally, the last group did their work in tissue culture cells in vitro. And they knocked down ATG16, the normal ATG16, and then expressed the Crohn's disease variant. And they found that the autophagy to bacteria didn't happen in the same way. It was less efficient. So suggesting that perhaps there could be inefficient autophagy in response to bacteria. So that's what was known about ATG16 in the initial discovery. So we were very interested because we knew that NOD2 was involved in inflammatory bowel disease and this autophagy pathway. NOD2 is an intracellular bacterial sensor, so we asked the question, is there an interplay between these two pathways to help to promote inflammatory bowel disease? And we wondered, is NOD2 involved in sensing a bacteria and initiating autophagy to help get rid of it? So we did this work. We collaborated with Dana Philpott at the University of Toronto, who's an international expert in NOD-like receptors. And we asked a very simple question. Does NOD2 ligands, so that component of the bacterial cell wall, does it trigger this autophagy pathway? So we took macrophages from wild-type mice that had normal NOD2, and we transfected these cells with LC3. So remember, I told you under normal conditions, LC3 is in the cytoplasm. So that's what you see here, this red. The blue is the nucleus. And when we incubated them with the NOD ligand, that component of the bacterial cell wall, we started to see all these autophagosomes, or those recycling bins. So you can see all these red dots here. These are autophagosomes. So is this because of NOD2? Well, we then looked at macrophages that didn't have NOD2, and they didn't have this response. So showing that it was specific to NOD2. And I just want to point out that this work was done by Seamus Hussey, who was a GI fellow at Sick Kids who worked with Dana and myself. So he did parts of this, this work. So it looks like the bacterial component is recognized by NOD2 and it's triggering autophagy. So how is this happening? How is it signaling autophagy? So we next wondered, does NOD2 and ATG16 associate? So does NOD2 sense the bacteria, and then does it bring this autophagic machinery to help ingest the bacteria? So to look at this, we took cells and we infected them with a pathogen, Shigella. It's an infectious organism that causes dysentery, horrible disease. So this is just looking at the surface of the cell. And you can see here these rod-like structures. This is Shigella invading into the cell and the cell taking up Shigella. So we then looked at where is ATG16, and we found that here you can see the bacteria in blue. ATG16 is coming to this bacteria. And what about NOD2? Well, NOD2 here is shown in green. NOD2 is coming to where this bacteria is as well. So the bacteria is coming in, NOD2 is there, and so is ATG16. So are they partnering together to induce autophagy? So if that's the case, what happens when you've got the NOD2 frameshift mutation? So remember, this doesn't recognize bacteria, it doesn't set off that inflammatory cascade. Does it affect the autophagy pathway? Remember, we're trying to bring together these two pathways in Crohn's disease. So again, here's the surface of the epithelial cell. Here's the bacteria coming into the cell. And this time when we've got the NOD2 frameshift mutation, there's no NOD2 where the bacteria are, and there's no ATG16 either. So if we don't have that proper sensing of NOD2, the first signal, bacteria comes in, NOD2 recognizes the bacteria, we're not getting ATG16 there. So are we not getting autophagy in that case? Well, in fact, no, we're not. So here's wild-type cells and cells that have the NOD2 frameshift mutation associated with Crohn's disease, infected with Shigella. We expressed LC3 in the cells so that we could follow it fluorescently. So here, Shigella is in red, and here are autophagosomes. They're rapidly wrapping the bacteria. So the bacteria is rapidly getting ingested by autophagosomes to control infection. But what's happening when we have the NOD2 frameshift mutation? Well, here's our cells, and there's really very minimal LC3 that is being targeted to those cells. And so really what this is showing us is that if you've got the NOD2 frameshift mutation, you're not recognizing the bacteria and you're not setting off that autophagy pathway. So this is a model of what we think is happening during infection with bacteria. So here's our cell. Bacteria is invading into the cell. It's being sensed by NOD2, which is interacting with ATG16 to trigger this autophagosome to ingest the bacteria and help get rid of infection. However, if we have the NOD2 frameshift mutation, this is not happening properly, and so we're not able to control infection with enteric organisms as well. So that's very interesting. The NOD2 frameshift mutation is affecting the autophagy pathway. We're linking these two pathways together. So what about ATG16? What is that ATG16 variant doing? So there was a paper that came out around the same time as our Nature Immunology paper that looked at blood cells from patients with and without the Crohn's disease variant of ATG16 and suggested that they had inefficient autophagy in response to bacteria. So again, suggesting that there's a link between these pathways. And if you have the NOD2 frameshift mutation, you have a defect in this pathway. If you have the ATG16 Crohn's disease variant, maybe you also have a defect in this pathway. So we wanted to further ask questions about this, and we really wanted to look in human patients. So do variants in ATG16 affect colonization with enteric bacteria and in patients? So that brings me to my other love in the lab, which is H. pylori. So let's bring it back to the patient again. So this is another 9-year-old boy who presents with a history of epigastric pain, and his family history is significant for inflammatory bowel disease in his mother. So he also had some nighttime wakening, so we did a scope because we were concerned maybe he had peptic ulcer disease. We wouldn't have done it otherwise if we thought he had functional abdominal pain. And so when we do the scope, we see in the lining of his stomach, right before the pylorus, there's all these bumps. So this is classic for Helicobacter pylori infection. And when we do a biopsy, we see the bacteria present. So this child has H. pylori infection. So what do we know about H. pylori infection? So it's a very common infection. So 50% of the world is infected with this organism. Much more common in developing countries, less common in developed countries. And this is just an image of the bacteria. So why do I care about H. pylori infection? Well, we actually acquire the infection in childhood. So you acquire the infection in childhood, and it persists with you for life unless you receive specific eradication therapy. Very, very unusual to have self-eradication. And the overwhelming majority of individuals infected with the bacteria will have inflammation but not have symptoms. About 10% of individuals will develop peptic ulcer disease, and less than 1% of individuals will develop gastric cancer. So the majority of people don't have disease, and some have said it may even act like a commensal organism. So a work done by a postdoc in my lab, Mauricio, identified that H. pylori not only can be extracellular, but it can actually invade into epithelial cells as well. And these are electron micrographs, so very high power images of gastric tissue from an individual infected with H. pylori. And you can see H. pylori in this vacuole, which is right within the cell. And this is another image showing you the same thing. Mauricio also looked at gastric cells just grown in tissue culture. We infected them with the bacteria, and they could invade and swim in these big vacuoles and live quite happily for up to 24 hours. So we wondered, since H. pylori can be invasive, whether or not it might interact with this autophagy pathway. And so we did some very simple experiments. We took gastric epithelial cells, we grew them in a petri dish, and we added the bacteria, and we saw what happened. We did very high power images, so these are electron micrographs. And as a control, to make sure we could identify autophagosomes, we starved some cells. And here you can see this is what those recycling bins look like in the cell. So you can see this walled off material, all kinds of stuff within the autophagosome. And when we infected our cells with H. pylori, here you can see the cells, there's a bacteria within this big vacuole, And we can also see these autophagosomes, or these recycling bins, with the bacteria within them. So infection with H. pylori is triggering this pathway in gastric epithelial cells. So we looked at it another way, and this was work that was done by Deepa, who's another postdoc in the lab, using that fluorescent probe labeling of LC3. So here's control gastric epithelial cells, and under normal conditions, the LC3 is just diffusely in the cytoplasm, so you can see it here, just this diffuse green staining. We can add a drug called rapamycin, and that triggers autophagy, just like starvation does. And now we can start to see these puncta, these recycling bins within the cell that are starting to self-digest the cell. And interestingly, when we infected the cell with H. pylori, we saw the same thing. So lots of these recycling bins or autophagosomes, and when we looked closely, we could see that some of those autophagosomes were actually ingesting the bacteria. So is this a host response to the bacteria to try to get rid of the bacteria? So if it is, what is it in the bacteria that's triggering this host defense? And I won't go through all the studies that we did, but we found that it was due to a specific toxin that's produced by the bacteria. So this is called the vacuolating cytotoxin. So not all H. pylori have this vacuolating cytotoxin, and it's associated with more severe disease. And so what does this toxin do? So it's secreted by the bacteria. Here it is here. It looks beautiful when you look at it in very high-powered view. It really does look like this little flower. It binds to the epithelial cell. It's taken up, and it causes the formation of those large vacuoles. So if you remember that picture I showed you of the epithelial cell with the big vacuoles and the bacteria in it, that's from this toxin. So we found that just this toxin alone could trigger autophagy in epithelial cells. So these results are showing that. Again, these are gastric cells. They have that LC3 that's got the fluorescent probe, so we can identify autophagosomes. So untreated cells, nice diffuse in the cytoplasm. When we treat with toxin that doesn't have activity, looks just like the control cells. But when we treat with active toxin, we start to see all these autophagosomes again. So all these little recycling bins. So are they trying to get rid of the toxin? Is it a host response to try to get rid of the toxin? So that's the question that we asked next. And so in order to answer this question, we took gastric epithelial cells, we incubated them with the toxin, and we let it sit there for 6 hours, and we washed it off, and we lysed the cells, and we ran them on a gel, and we measured the toxin over time. So this is using an antibody to VAC-A. And we compared that in normal cells with cells that didn't have autophagy. So this is shown here, these cells, they can't induce the formation of these recycling bins. And in the normal cells, if we look over time at what happens to the toxin, you can see over time the toxin's getting degraded. So we just kind of see a mush here in this last band. So the toxin's getting eliminated. But if we don't have autophagy, and we measure the toxin over time, it's persisting. So showing us that this is actually a host response to try to get rid of that toxin. And we can look at that another way by looking at how the cells still have vacuoles over time. So remember, this is what the toxin does. It induces the formation of these big vacuoles. So in control cells that we've washed off the toxin, over time, by 24 hours, no toxin, right? There's no toxin here. We don't see vacuoles anymore. When we look at the autophagy deficient cells, at 24 hours, we've still got lots of vacuoles. And that's because, as you can see here, we still have toxin. So showing us that this autophagy response is happening in the cell, and it's trying to get rid of the toxin. So what we think is that during infection with H. pylori, the cell is sensing the toxin somehow, inducing autophagy. That's decreasing the presence of the toxin, which we'd shown is important for promoting colonization. So it decreases bacterial colonization, potentially limits intracellular bacterial survival. So really, maybe this is a very important host response to infection. So when I started telling you about this, I said what we really wanted to do was to look at whether or not ATG16 can actually have an effect on infection in humans. So we asked the question, can autophagy modulate H. pylori infection? And so to do that, we did genotyping in a case control study. So we collaborated with a geneticist in Scotland who had this very interesting group of individuals who were first-degree relatives of somebody that had gastric cancer. So first-degree relative, same environmental situation. H. pylori is the single most important risk factor for the development of gastric cancer. So these individuals would have been exposed to H. pylori infection, and what you can see is that some of them developed H. pylori infection and some of them did not. Now they characterized them very well into whether or not they had changes associated with pre-malignant changes in the stomach or not. That really doesn't matter. As controls, we used cord blood samples from individuals from the same region in Scotland, and we genotyped them for ATG16. So did they have the wild-type ATG16 or the Crohn's disease variant? And what we found was very interesting. So when we looked at the controls, remember at the beginning I told you that about half of Caucasians have this variant? About half of the controls had the Crohn's disease variant. That's the G variant. When we looked at the individuals infected with H. pylori, remember we were thinking that this ATG16 Crohn's disease variant results in deficient autophagy. We think autophagy is an important host defense against the bacteria. And we found that in the individuals that were infected, so they were susceptible to infection, they actually had a higher proportion of the Crohn's disease variant. And interestingly, when we looked at those that weren't infected compared to the controls, they actually had a higher frequency of the wild-type variant. So we think this is pretty compelling evidence that this Crohn's disease variant in ATG16 alters susceptibility to infection. And we're not happy just to do one case control study. So we wanted to replicate this because maybe that just happened by chance. And so we did another study in the German population, Caucasian individuals, those that were infected with H. pylori and those that weren't, and we found the same thing. So there was an increased frequency of the Crohn's disease variant in those individuals that were infected with H. pylori. So autophagy is a host defense against H. pylori infection. I showed you some in vitro data to support that. And this is genetic studies showing that if you have a variant in an autophagy-dependent gene, which we think results in less efficient autophagy, you have more susceptibility to infection with a bacteria. So this just summarizes what we think is going on during infection with H. pylori and may have some implications for inflammatory bowel disease as well. So if you've got the wild-type ATG16, when you have infection with H. pylori, you've got normal autophagy, which helps to decrease the toxin and limit intracellular survival. However, if you've got the Crohn's disease variant, this autophagy is less efficient, and this may allow the bacteria to survive intracellularly, increase VAC-A, and promote colonization and chronic infection. So I hope what I've showed you is that autophagy isn't just a recycling process. It's involved in immunity, it's involved in the microbial interface, and disease pathogenesis. In particular, I told you about two gastrointestinal diseases and its role, Helicobacter pylori infection and Crohn's disease. There's also a lot of interest in the role of autophagy in malignancies, and my lab's working on this as well, but I didn't have time to tell you about that information. There's also other gastrointestinal diseases that it's been implicated in, and many of the diseases that you're interested in, there could be a role for autophagy as well. So why is this important? Why do we need to understand this pathway? Well, what we're hoping is if we understand this pathway, we can develop therapies that can impact on this pathway. And this is actually happening here and now. So right now there's over 30 trials, clinical trials that are registered looking at drugs that impact on the autophagy pathway for different diseases. And these are just some examples of some studies that are demonstrating this. So this paper in Science was a very interesting paper which looked at a disease called alpha-1 antitrypsin deficiency, which can cause liver disease in children. And it's because an abnormal protein is made. And so autophagy can also get rid of those abnormal proteins. And so this was studies in an animal model where they increased autophagy, got rid of that abnormal protein, and impacted on liver disease in mice. But they're now doing studies in humans looking at this effect. Autophagy is implicated in cancer, and there's a lot of interest in trying to develop agents that can impact on this pathway to be used as chemotherapeutic agents. I told you about inflammatory bowel disease and whether or not we can impact on this pathway to help treat inflammatory bowel disease. There's only one paper that's looked at this, but I think this is an emerging area. And so with that, I'd just like to acknowledge the people in my lab that have done the majority of this work. I'd like to acknowledge our collaborators, our funders, and importantly, I also want to thank the Canadian Association of Gastroenterology, which helped to sponsor my visit here to you today. And with that, I'm going to close, and I'm happy to answer any questions that you have.
So maybe just before the questions, I have to add to the thank you. So Nicola is the visiting research professor for the Canadian Association of Gastroenterology, which really stimulated her visit to here. But the visit was also supported by the Walter Mackenzie Visiting Lecture Fund and by WICRI. So thank you to both of those institutes. And why don't we start with the remote sites first? Are there any questions from the remote sites? Okay, let's move to questions from here, please.
Not quite remote, but
well, you're far up there.
I want to thank you for that. That was a really fascinating talk. What I was wondering if you could do is connect the issue of the granuloma with the function of the ATGL16 and the NOD1, because granuloma formation is abnormal. So why are we seeing abnormal cell progression? What's happening with cell cycle progression in aberrant autophagy that's different?
So we don't have any, oh, sorry. So the question was what's happening in granuloma formation. So if you remember at the beginning of the talk, I showed you a granuloma in a biopsy specimen, which is characteristic of Crohn's disease. And so the question was whether or not autophagy could play a role in the formation of the granuloma. I think I'm rephrasing the question. And so we don't have evidence that it's involved in granuloma formation, but that's only from genetic studies that have looked at whether or not there's any association with genotype ATG16 and granuloma formation, and there's not evidence for that. But whether or not it could be involved, I'm not sure. So I think really we need to have an in vitro model system to look at granuloma formation and see how autophagy would impact on that.
It's just that when you show the pictures of autophagy being induced by rapamycin, it's a very compelling connection to want to make something that's changing cell cycle progression.
Right. So yeah. So rapamycin inhibits mTOR, which is implicated in autophagy. Rapamycin does a lot of other things that aren't autophagy related as well. So how autophagy affects cell cycle and cell death is an area where there's a lot of controversy, actually. So I can't give you a specific answer about that.
Yes.
Thanks for asking. So we have a lot of Crohn's patients have polymorphism in NOD2 and ATG16 that don't have the disease expression. So like 60 people have population have that. So have you looked at, say, commensal and this pathway to see, why does some patient that get Crohn's and others don't? And yet in your model too, you show that, the exposure happens and affect this pathway, connecting the NOD2 polymorphism with ATG16.
Yeah, so the question is, there are individuals that have these variants. So I told you that half of the population can carry a copy of ATG16 Crohn's disease variant. So why is it that some people get Crohn's disease and some people don't? And I think it comes back to that model where there are many factors that are involved. So it is a multifactorial disease. And so having autophagy deficiency may just be one part of that puzzle. And there may be compensatory mechanisms in normal individuals that don't make that become an issue. But under exposure to the right environments, perhaps infection sets off that pathway, and if it's not sufficient, then that may set forward the cascade of disease. So we don't really know, and that's obviously a speculation, but I think in any multigenic disorder, it's clearly having that variant is not sufficient to cause disease, but it's part of the puzzle. So there are other factors that are involved. And that's really why we wanted to look at NOD2 and ATG16 together to try to start to piece together some of these pathways to see if there is some commonality to them.
Todd?
I echo my colleagues' thanks for a beautiful picture and beautiful talk. And I thank you for making the immune system even more complicated. So with respect to that, you're showing very clearly Shigella on the cell surface, and that we're not getting autophagy, you're getting that in lack of immunity. Two questions, I guess. One is, do these patients with this frameshift mutation at any increased risk of invasive disease? Or is this one small piece of the immune system and you've got all these other mechanisms so they aren't really at an increased risk, and this is just setting them up for these subtle diseases, these other diseases that are subtle?
Yeah, so I think, so to repeat the question, so I showed that the NOD2 frameshift variant affects autophagy in cells. And so are individuals that have this variant susceptible to infections, or is there some compensatory mechanisms? And I think that that's probably the case. So we don't really have specific evidence that those individuals are immunodeficient, although there are some interesting in vitro studies looking at immune responses from peripheral blood cells, for example, that show that there is potentially a defective immune response in inflammatory bowel disease. And work by one of my colleagues who's looking at early onset inflammatory bowel disease. So inflammatory bowel disease that develops in children less than 6, for example. And what are the genetics behind that? A lot of the genes that he's identifying are associated with immunodeficiency. So there may be an interplay, but I do think there are compensatory mechanisms. So autophagy is not the only way that you can eliminate intracellular pathogens. There are other mechanisms. So presumably it's a multifactorial process that you have this interplay that it has an effect on somebody with inflammatory bowel disease. Similar to what I told Hien, I think that there's multiple factors that have to come together, and just having the NOD2 frameshift mutation isn't sufficient to cause disease. So there's other factors that are involved. So this is just one part of the puzzle.
I might ask a question. So autophagy is fascinating, and really the attention it's gotten over the last few years has been tremendous, but it really seems like it's a very fundamental biological process involved in housekeeping within the cell. And I wonder if there are basic factors and external environmental interventions that will manipulate the autophagy pathways in a much more immediate and simple way than using drugs. So starvation in yeast cells, I know, has been shown to be the most simple one, but has anyone looked at the effects of nutrition or different exposures on the tone of autophagy, and is that something that might relate to complex conditions such as IBD?
Yeah, so the question was, let me just try to see if I can rephrase this. So autophagy is a very fundamental process. I showed you it's important for cellular homeostasis. So rather than have drugs that specifically target this pathway, are there ways that we can manipulate it, such as with nutrition, for example? And so So definitely there are some studies looking at the impact of nutrition on autophagy, the impact of vitamin D therapy even. And not to become overly complicated, but there are different triggers for autophagy. So you can have non-selective bulk autophagy, which probably happens in cellular homeostasis. But there are also specific proteins within a cell that will target material to be degraded into autophagosomes. So we know, for example, in bacterial infection, they can be targeted by a specific adapter protein that will trigger autophagy specifically for that bacteria. So rather than the whole cell just ingesting itself, it can be targeted to remove specific material. So it may be, if we identify where autophagy might be deficient in inflammatory bowel disease, that we could target that pathway specifically by perhaps manipulating some of these adapter proteins as well. So I think your point of nutritional therapy is very interesting, and there are actually some people in Canada that are starting to look at that in inflammatory bowel disease. So yes, important question.
More questions from the audience or from the remote sites? Okay, well, I think we're just on time, so thank you so much again, Nicolas.